Welcome to This Explainer. Today, we're mastering the absolute foundation of biological research, the scientific method. We're going to lock down the exact, clean, step-by-step rules of experimental design that you absolutely need to know to ace your standard textbook board exams.
Okay, I'm going to interrupt you right there because look, while rote textbook memorization is a fantastic starting point, we really need to look at how those pristine rules hold up against the messy, unpredictable reality of actual biological lab work. I want to show you what happens when theory actually meets the benchtop.
Okay, let's dive right into this. The textbook method gives us a really beautiful, clean sequence, right? You make an observation, you form an educated guess, you run an experiment, and boom, you prove your hypothesis is correct. It's a very satisfying checklist.
Oh, it is definitely satisfying, but honestly, it's also a bit of a myth. Here is the actual lab reality. Science never actually proves anything. Real-world research is entirely about controlling for chaotic variables and trying to reject the null hypothesis. It's way less about finding absolute universal truth and much more about slowly and carefully eliminating what is false.
Section 1. Hypotheses and Experimental Design.
Before you even think about touching any lab equipment, you have to establish your hypotheses. The strict textbook definition of the null hypothesis, which is often written as H0, is that the conditions being looked at or tested will have literally no effect. The word null literally translates to nothing. It represents the absolute baseline assumption that nothing is happening and there is zero relationship between your variables. So here's a question for y'all. If you run your biological experiment and you see a massive, undeniable effect, did you just prove that your experimental idea is true? How does a real lab researcher actually interpret a successful outcome? Well, the harsh reality is this: ideas just can't be proven. They can only be rejected. In the lab, a currently held belief is essentially just an explanation that happens to fit the data best, right up until a better explanation comes along. Science proceeds very conservatively. So, when your experiment works, you haven't proven your idea at all. You've merely successfully rejected the null hypothesis. Let's move on to Section 2, Variables, Groups, and Controls. Let's look at how we actually set up an experiment to test these ideas using a classic textbook example like measuring the growth of peanut plants. For any board exam, you absolutely must be able to identify essential variables. First up, the manipulated or independent variable. This is what the investigator deliberately varies, like the exact amount of fertilizer applied to the plants. Second, the responding or dependent variable. This is what you actually measure, like the height of those peanut plants. Then you've got controlled variables, which are factors kept perfectly equal in all treatments, like the type of soil or the brand of fertilizer, just so they don't influence the outcome. And finally, you set the levels of treatment, like applying exactly 200, 400, or 600 grams of fertilizer. And see, this brilliantly illustrates the lab reality. Because just knowing your variables isn't enough. You must have experimental controls. A positive control is a sample treated in a way that is absolutely known to produce a positive result. A negative control is treated exactly the same as your experimental samples, but isn't expected to change at all. For instance, in a real-world Western blot looking for a specific protein, your positive control is a cell lysate you know for a fact expresses the protein. Your negative control is a lysate that definitely doesn't. Listen, if your positive control fails, or your negative control magically lights up, your whole experiment is void. It is a soul-crushing day at the bench. You absolutely need these to ensure your reagents are actually working and your results aren't just weird artifacts.
Section 3. Data Quality and Error Analysis.
When you measure a dependent variable, you need to rigorously evaluate the quality of your measurements. You're almost certainly going to be tested on the difference between these two terms. Accuracy is how close your collected values are to the real, true value. Precision is how close all your measurements are to one another, totally regardless of whether they are near the true value.
Right, and out in the field or at the bench, achieving both requires meticulously calibrated equipment. If your pipettes are slightly off, you might have incredible precision, meaning your measurements are tightly clustered together, but you'll have terrible accuracy because every single drop is just wrong. A solid, real-world experiment requires proper, maintained equipment to capture data that is both accurate and precise.
Section 4. Descriptive Statistics and Variance. Descriptive stats like the mean, median, and mode help us pinpoint the center of our sample. But to truly understand the spread of the data, we use standard deviation.
You calculate it by finding the difference between each data point and the mean, squaring those differences, averaging them, and taking the square root. For data with a perfectly normal distribution, roughly 68.3% of the data lies within exactly one standard deviation.
Now, what's really fascinating about this is how it applies to messy biological populations. Biological data is rarely perfectly uniform. Whether you're measuring the territory size of little green bulls in a forest or the percent weight gain in a group of guinea pigs, your data is going to spread out. Knowing that roughly 68% of your real-world variance is captured within that first standard deviation helps you instantly visualize just how chaotic or uniform your test subjects actually are.
And that brings us to Section 5, Inferential Stats and Significance.
Descriptive stats summarize the sample, but inferential statistics allow us to actually predict parameters for the entire population. For your exams, you need to memorize this matrix. Parametric tests, like the independent t-test or ANOVA, are used for normally distributed data. Non-parametric tests, like the Mann-Whitney U or Kruskal-Wallis, are used when data just doesn't fit those clean assumptions. And here's how a researcher actually chooses a test in the real world. In a textbook, data is often perfectly normal. But in biological field data, samples are frequently way too small to be confident about the shape of their distribution, or there are totally natural reasons the data is heavily skewed. When primatologists or ecologists are out in the field, they're constantly forced to rely on non-parametric tests that compare medians instead of means, precisely because nature just doesn't neatly fit a parametric bell curve.
Now, regardless of which test you run, it will generate a probability, or a p-value. The classic threshold for statistical significance is p less than 0.05. This is the absolute standard benchmark. If your p-value is less than 0.05, it's considered sufficiently small that we conclude the effect is not due to chance, and we confidently reject the null hypothesis.
Whoa, let me stop you right there, because this is probably the single most misunderstood concept in all of lab science. P is not the probability of the null hypothesis being true. So the totally crucial point here is that a p-value of 0.05 does not mean there's a 5% chance the null hypothesis is correct. It actually means that if the null hypothesis were entirely true, there is less than a 5% probability of obtaining the exact data you just collected. It is a subtle but absolutely massive difference in how we view truth in science.
That is a fantastic clarification. The textbook definitions of variables, controls, and p-values really give us the rigid structure we need to ensure our experiments are valid and our board exams are passed with flying colors.
But as we've seen today, actually applying those textbook concepts to a messy Western blot or a skewed population of field subjects requires deep critical thinking. Science isn't about perfectly proving your first guess. It's about rigorously attempting to reject it until only the absolute most resilient explanations remain. So we're going to leave you with this final thought. If the textbook provides the map, are you ready to navigate the unpredictable, wild terrain of the biological laboratory? Think about how you'll apply these structured rules to your next major scientific discovery. Thanks so much for joining us for this explainer.